COVID-19 has had a significant impact on our world over the past few years. Now, of course, the most direct and tragic impact is through the number of lives that have been lost due to this pandemic. But even those that have recovered from COVID-19 will also experience long-term effects from their illness. Additionally, we have larger societal changes, such as an increase in mental health concerns, as well as changes in education due to COVID-19 precautions. Because of the significant impact of COVID-19, the mechanism of COVID-19 infection has been an area of intensive research over the past few years as well. As researchers have investigated the mechanism of COVID-19 infection, they found that carbohydrates have a key role in the infection process. So COVID-19 binds to our cells through this ACE2 protein receptor, which you might have heard about in various COVID-19 news articles. But that interaction is also strengthened by an interaction to a sugar polymer, our heparan sulfate. Heparan sulfate is a glycosaminoglycan, which is a heteropolysaccharide that is highly negatively charged that can interact with our virus particle. So as you can see, our virus particle interacts with that heparan sulfate marked here, and that interaction then helps to facilitate the interaction with the ACE2 receptor, marked here with the brown color, before eventually leading to infection and viral replication within our cell. This interaction was studied specifically using absorbance spectroscopy. So as you can see in the graph down here, so they added different concentrations of the spike protein, and they looked at the absorbance after the protein bind to a surface that was coated with either heparin or the protein BSA. As you can see, our absorbance increases in the presence of the heparin as the spike protein binds to that heparin. So of course, we could use this to determine a KD, and actually look at the interaction between that COVID-19 spike protein and our heparin quantitatively. This binding interaction between the COVID-19 spike protein and heparin is directly responsible for the infection of the cells. So in order to investigate this, the scientists performed a mock infection protocol, and they saw roughly 40% of their cells, marked here on the y-axis, were infected under their mock conditions. They then added different amounts of free heparin here, marked here as UFH. So if you're adding free heparin, that heparin will compete with the binding site of the COVID-19 spike protein compared to the heparin that's linked to the membrane through that whole glycosaminoglycan structure. So it's competing with the heparin on the cells. And what we see is that when that free heparin is present, increasing concentrations, it decreases the infection rate. So essentially, it's making it harder for the spike protein to bind to the cells and then ultimately infect our cells. They also added an enzyme that would break down the heparin so that, again, that would reduce the binding of the spike protein. And again, they saw that the infection rate significantly decreased. So as I mentioned previously, a heparan sulfate here is a highly negatively charged sugar polymer. And it will bind to positively charged amino acids. In this case, it's binding to amino acids from both the spike protein, and then it's helping to cross-link that to the ACE2 receptor as well. So as we saw with that data on the last slide, this sort of interaction can help to increase the interaction between the proteins and then eventually trigger things such as infection. So carbohydrates, through these interactions with different proteins, actually play a critical role in signaling within our cells and even in cases such as viral infection. The key sugar polymers that are involved in this sort of cellular signaling are glycosaminoglycans. Glycosaminoglycans are linear heteropolysaccharides. So again, that means that they don't have a lot of branching, but they will have different sugars combined to make the polymer structure. So most of our glycosaminoglycans will have repeating disaccharide units. So we'll have two unique sugars, but then we'll repeat those same two unique sugars over and over again. For every glycosaminoglycan, we're going to have one monomer that's an N-acetyl sugar. So notice here we have the glycosaminoglycan hyaluronate or hyaluronic acid shown, and it uses the glucose N-acetyl modified sugar. So of course the N-acetyl group, as we talked about before, slightly modifies the reactivity of our sugar monomer here. The other key component of our glycosaminoglycans is some form of negatively charged sugar. This could be an oxidized uronic acid sugar, or it could be a modified sulfate ester sugar. Again, this is just different ways of introducing negative charge in order to make our sugar better able to interact with proteins that might have positive charges, right? So those sort of charge-charge interactions are some of our stronger intermolecular interactions. So it's helpful to have charge groups on our molecules that we want to be interacting with a lot of other substrates. So notice with our hyaluronic acid here, we have our glucose oxidized in order to produce our negatively charged carboxylate group here. Now, the other thing you'll notice here about our glycosaminoglycan is that we actually do have two different glycosidic bonds here. So we have a beta 1 to 3 and a beta 1 to 4 bond linking our sugars together. So this is a unique case where we have two different glycosidic bonds, but it's not resulting in branching, right? It's just we're varying our bond up as we go along our linear polymer. The sugars that we use in our glycosaminoglycans will vary significantly between different kinds of glycosaminoglycans. So here we have chondroitin 4-sulfate, and you'll notice now we have galactose here instead of our glucose, and we also have an additional sulfate ester added on here as well. So the key things that stay the same will almost always have an N-acetyl sugar, and we'll always have some negative charge here, but the exact sugars will vary depending on what glycosaminoglycan we're looking at. Now, of course, heparin is a polysaccharide that we're interested in for COVID-19. If we look at the structure of heparin, you'll notice again we have a amine modified sugar, although in this case it's just a single amine group and not the N-acetyl group. And of course, we also have a lot of charge groups on our glycosaminoglycan here as well. So our heparin is actually the highest negative charge density biomolecule that we have, based on all of these negative charges from our various sulfate esters, as well as that carboxylate group. So heparin is just the polysaccharide alone, and then our heparin sulfate is when that heparin is attached to our protein to make our full glycosaminoglycan structure that we saw involved in that COVID-19 infection. Here's a representation of our heparin. So notice all of these reds here from our oxygens that are also going to have a high degree of negative charge. And so our heparin will tend to form a very extended structure where we can make a lot of interactions with proteins as our negative charges are kind of repelling each other and spreading out our heparin, and then making it available for binding to our positively charged proteins. And again, as we saw with COVID-19, our heparin can help to bridge between positively charged regions of different proteins and increase the interaction between those two proteins. So heparin is also involved in the interaction between thrombin and antithrombin. So this allows the antithrombin to inhibit our thrombin protease and prevent blood clotting. So this is involved in, of course, preventing our blood from clotting up as it's flowing through our body. And then it's the removal of our antithrombin that then will occur as we're going through our blood clotting cascade when we actually have an injury. It also will bind to various cells, and it can regulate development and formation of blood vessels. And as we saw, it's also involved in interactions with viruses and bacteria. While our glycosaminoglycans perform many important roles on their own, in many cases they'll be linked to other biomolecules in glycoconjugates. So a glycoconjugate is a biologically active molecule where we have a carbohydrate linked to either a protein or a lipid. These glycoconjugates can occur in many different forms. So we can have varieties of glycoproteins and proteoglycans, which are combinations of carbohydrates with proteins, or we can have a variety of different glycolipids with those carbohydrates linked onto lipid molecules. A glycoprotein is a protein with a small oligosaccharide polymer attached onto your protein amino acid. So of course, we need to be able to form some kind of bond between our sugars and our protein. This bond will occur as a form of a glycosidic bond between our anomeric carbon, so that would be on our sugar chain, to one of our amino acids. So we'll commonly see it attached to serines, threonines, and asparagines. So our serine and threonine, of course, can use the alcohol of the side chain to form a normal glycosidic bond with the anomeric carbon of our sugar, right? So notice how we formed a normal acetyl here with our alcohol acting in the same way as another alcohol from a sugar would. So in this case now our sugar has been linked onto our protein polymer instead of onto another sugar molecule. So we refer to these as O-linked bonds because it's attached to the oxygen of either our serine or our threonine. An alternative is an N-linked bond. So here we form what we call an N-glycosidic bond, where now we've linked to our acetyl, but in this case, we're using a nitrogen as our nucleophile to attack that hemiacetal rather than our oxygen. So this nitrogen will most commonly come from an asparagine, although it could come from other amine side group amino acids as well. These glycoproteins are actually quite common in mammalian systems. So we see about half of mammalian proteins are actually glycoproteins, again with those sugars, polymers attached in either O-linked or N-linked configurations. They'll play a key role in protein-protein recognition, right? So as we saw with heparin, right, those carbohydrates are very useful for interacting between different proteins and helping to bring those together. So that will also be a key role here with our glycoproteins as well. Because those glycoproteins are so important for protein-protein recognition, viral proteins will also tend to be heavily glycosylated. So that'll allow them to kind of look like the normal glycoproteins that we might see in our bodies in order to try to evade the immune system. Proteoglycans are a specific type of glycoconjugate that uses sulfated glucosaminoglycans attached to proteins which are linked to the membrane. There are multiple different kinds of proteoglycans. So two of the main kinds are syndicins and glypicans. So syndicins use a protein domain that's actually embedded in the membrane. So we refer to this as an integral membrane protein. And our glycans are actually attached to the membrane through a linker. So we have this GPI anchor, it's called, that will link our protein to the membrane. And they both will have those sugars attached. So we see here in green, this is our heparin sulfate. So we saw, of course, the structure of heparin before. Heparin can be linked to our protein as our heparin sulfate to create these proteoglycans. Proteoglycans play a key role in the interaction between cells and the extracellular matrix. So the extracellular matrix, of course, is a key component of our tissues, right? It makes this kind of external scaffold that helps to hold our cells and our tissue together within our organs. The extracellular matrix itself is formed from fibers of protein. So we have collagen here. We also have our proteoglycans with those carbohydrates here, and other proteins such as fibronectin. The extracellular matrix will then interact with a variety of proteins from our cells. So we have integrins, which are one form of proteoglycans, as well as our different kinds of syndicins. The glycosaminoglycans used in these proteoglycans have a variety of different roles. First, as we've seen already, they can enhance interactions between proteins, right? So we saw previously how the binding of antithrombin and thrombin could be increased through this sort of cross-linking with our heparin. So our heparin is marked here in green. It can bind to our antithrombin and then bind also to our thrombin and help to bring those into close proximity where that interaction will be strengthened. Some of the other mechanisms that our glycosaminoglycans will use to increase protein interactions are through conformational activation. So we might have a protein that will bind to our heparin and then be activated by switching into a separate conformation to then increase the binding to its binding partner. In addition to enhancing protein-protein interactions, like we saw with thrombin and antithrombin, we can also see these glycosaminoglycans used as co-receptors for extracellular ligands. So here we have an interaction between a ligand and its receptor, and we see our heparin sulfate here in green used to, again, bridge between that ligand and receptor and help to increase the strength of that interaction. In some cases, they may also be used to increase the concentration of ligands near the cell surface. So if we have particular ligands that will bind to our glycosaminoglycans, that will then increase the relative density of those molecules near our cell surface. So that can then allow them to, again, increase their interaction with other molecules because we have a higher relative concentration there near our cells. So all of these are different ways that our proteoglycans will use those carbohydrates in order to increase various interactions between our cells and other binding partners. In addition to linking our sugars to proteins, we can also link sugars to lipids. So we'll form a variety of different glycolipids. These are lipids with covalently bound oligosaccharides. And of course, we'll look at our lipid structures in more detail in a later section of our course. But for now, you can understand they're going to be using similar glycosidic bond linkages to our sugars. So here we have an alcohol from our lipid being used to link to a large sugar polymer in order to create our glycolipid here. These will act as key parts of both plant and animal cell membranes. One of the very important roles of our glycolipids is for determining blood type. So our blood type is actually determined by the exact identity of sugars attached to these glycosphingolipids in our cell membranes. So you'll notice we have three different glycolipids here. We have one that is the O antigen, one that is our A antigen, and one that is our B antigen. And the difference here, you'll note, is just in what sugars are attached to our different lipids here, which you can notice as these little colored symbols here. So we have this additional sugar attached on for the A antigen, and we change which type of sugar is attached for our B antigen here. The production of these different kinds of glycosphingolipids is all based on what enzymes we have available. So we'll have specific glycosyl transferases that are involved in adding these sugar groups onto our glycosphingolipids. If we have no active glycosyl transferase, we won't add that final sugar on, and we'll end up with our O antigen up here on top. If we have an N-acetyl-galactosamine transferase, then we'll place that N-acetyl-galactosamine, which is represented by our yellow square, and we create the A blood group. If we have a glycosyl transferase that transfers just galactose, represented with our yellow circle here, then we end up with our B blood type based on this B antigen. So all of our different blood types are really just different varieties of glycosphingolipids within our cells. So these glycoconjugates, whether they are glycolipids or our glycoproteins, will have a huge role in interactions between our cells and various other cells or biological particles. So we see that these are involved in virus interactions with our cells. They're involved in interactions with bacteria, as well as with our leukocytes in our immune system. The key proteins that bind to these oligosaccharides are called lectins. So you'll notice for each of these different interactions that I highlighted earlier, we have a variety of different proteins that are binding to those carbohydrate chains listed in red. So we'll have different varieties of lectins used for binding to different carbohydrates. So notice we have P-selectin as our lectin here, binding to that carbohydrate of our leukocyte. This P-selectin is critical for targeting our white blood cells to sites of infection. So when we have an infection, of course, this leads to inflammation, and that inflammation triggers our P-selectin to move to the walls of our blood vessels, where then it can bind to glycoproteins on our white blood cells. So this will end up tethering our white blood cells to the surface here. It will then end up forming other interactions between our integrins and our white blood cell and the cells here of our capillary endothelial layer, and will eventually allow our white blood cell to invade and clear up that infection. As we saw earlier with the coronavirus, these sorts of protein-carbohydrate interactions can also be exploited to help viruses invade cells. So in many cases, our viruses are using varieties of receptors, often lectins, to bind to carbohydrates as they target our cells. So many antiviral agents actually use carbohydrate-like structures to target these lectins on the viruses. So on the left here, we see N-acetylneuramic acid, which is the target of the influenza lectin. This is found in many of our glycosaminoglycans, and it will then allow the lectin to target it and then lead to the virus invasion. So these two antiviral agents look similar to that N-acetylneuramic acid, so they can bind in the same binding site and prevent this sort of invasion of the virus by essentially blocking the binding to the normal carbohydrates of our cells. One of the things that makes lectin-carbohydrate interactions so important in our biochemical systems is that they tend to be highly specific and strong interactions. So within our sugar, we'll have generally many different sites where we can interact, right? So we'll have often those sulfate groups or other charged groups from our oxidized sugars that can bind to positively charged groups within our protein. So notice here we have a manganese ion that's helping to interact with the negative charge here. And then this is also interacting with various hydrogen bond donors of our protein backbone. We also, of course, have each of the alcohols on our sugar molecule that can form hydrogen bonds to various amino acids here as well. And so all of these interactions can kind of work together to create this highly specific interaction here to our carbohydrate. We can actually also use the hydrophobic effect here to help strengthen these interactions. So for many, our sugars will tend to have one side that's more polar and another side that's less polar. So if you look at our sugar here, you see a lot of our alcohols are kind of pointing up, including the carboxylic acid here for this oxidized sugar. And so these will all interact with hydrogen bond donors or acceptors on the top half of our binding site, while on the lower half of our sugar here, we have mostly our hydrogens pointing down to create a more hydrophobic face of our sugar. So here actually we're interacting through more of a hydrophobic interaction with our tryptophan side chain with that aromatic ring. So we can combine these along with the hydrogen bonding and charge interactions that I showed on the previous slide to create, again, that highly specific and very strong interaction to our carbohydrates. So what we see here in this lecture is that our sugars are far from just simple energy storage molecules or simple sugars that we use for fuel. They're actually incredibly important for interactions between cells, interactions between proteins, or interactions between lipids and other cells or lipids and proteins. So these sort of glycosaminoglycans and sugar polymers have an incredible variety of different shapes and sizes that we can make through all the different glycosidic bonds, as well as the different modifications that we can make to our sugar molecules in order to create these highly specific interactions between our lectins and their various sugar binding partners. We also again see that they're often used in combination with other biomolecules, right, through those glycoconjugates of our glycolipids or our glycoproteins. So sugar molecules are actually quite complicated and quite important components of our body. Carbohydrate biochemistry remains a very active area of research. Again, as we saw with our coronavirus, right, it's very important that we understand those sorts of interactions that are occurring between viruses and our carbohydrates so that we can gain better methods of targeting and preventing those sorts of virus infections, as well as just in general, understanding better how the biochemistry within our body actually works.